Says we're live. So anyway, as I was saying to you guys, after all that had passed and they'd all put their clothes back on, the fox turned around to the chicken and he said, oops, sorry, everybody's listening. I'll tell you the rest of the joke later. Anyway, people, we are here with a hangout. It's the Science Bitches Rogue Edition. For one reason and one reason only, we want to find out, what does the fox say? And CRISPR will explain what this is all about, because CRISPR has been dealing with this for a bit longer than I have. I sort of tuned in earlier on in the day today and very quickly lost the will to live. So CRISPR will explain this while I put my camera off, sit back and weep quietly. Off you go, CRISPR.
Yeah, so looking at the timestamps on the thread, I see that this was 20 hours ago, apparently. Tommy had posted a link to a little couple popular science things about an interesting case of a population of foxes that were developing a strangely high incidence of tumors, all in the ears. And apparently, some scientists had found that there was a correlation between infection with ear mites and the ensuing damage caused by those ear mites and the development of these tumors. And so they thought we should try treating these animals for ear mites and see if that doesn't help. And they found there's some positive results, that the tumor incidence seems to have dropped in the treated animals, which is great, because this is a very endangered population. There's not that many of them left, because almost all of them were wiped out by an outbreak of canine distemper about almost 20 years ago. So Tommy takes all of this in his usual fashion to mean that it's all environmental. There's no genetic element of this. It's just the environment. And because, in fact, that the fox's ears, it's protecting it from all the damage caused by the ear mites. Let's see, I have the quote here. Indeed, a protective mechanism generated by the body and put in place to seal off infection, dead cells, lactic acid from the rest of the body. That, of course, ignoring the fact that these tumors are found in a population of foxes on one island out of six islands in the same area that all have these foxes on them. And of those 6 islands, 3 of them have very high levels of ear mites, and only one of those has actual tumors. So apparently he thinks that it's just the environment, just the ear mites causing it, but only one group with ear mites gets tumors. Who knows? So I asked him, again, 20 hours ago, what happened to the treated animals that already had tumors, and why didn't animals on the other islands with the same incidence of mites get tumors? And that's when things rapidly started falling apart. So I don't want to read through 20 hours worth of other comments, but...
No. So to sum it up, let's just look at this. We've got some islands with some foxes on, and the populations were almost wiped out by canine distemper. The populations have come back. And then it's noticed that on one island, there's a whole bunch of foxes with little tumours in their ears, and they have ear mites. But on the other islands, there are foxes. They also have ear mites to the same level, but they don't have tumours. So, one island, ear mites, foxes, tumours. Other islands, ear mites, foxes, no tumours. What's causing the tumours? According to Tommy, it's the ear mites.
And just to share the diagram from the article, you see these 3 islands here have a lot of ear mites. almost 90+% infection rate with ear mites. And these islands here have the same, roughly the same foxes on it, no ear mites. Only this one has tumors.
Yeah. So why, if ear mites are the cause of the tumors, I'm going to actually just present you to all at the moment with that map. So if the ear mites are the only cause of the tumors, why don't San Nicolas and San Clemente Islands have tumors? Because they've got ear mites and foxes. Why is it only Santa Catalina that has ear mites and tumors if the ear mites are responsible? So from this data, you cannot say ear mites cause cancer. There's something else going on there that's singling out the Santa Catalina population. So what is unique to that population that allows heavy infestation of ear mites to give rise to cancerous tumours? Now, at the risk of using my favourite, this isn't rocket surgery, you can see what the conundrum is here. It can't just be the ear mites. There has to be something else going on. It's that simple. If it was just the ear mites, you would be getting these tumours on all 3 islands that have got ear mites. But you don't. Now, I know I'm belaboring a point that everybody in the audience is bound to have picked up on and is now saying, why does she keep going on about this? We all get it. It's because this is what seems to be getting missed in this thread that we've been involved in. If it was just the ear mites cause tumours, all 3 islands would have foxes with tumorous ears, and they don't. So why just Santa Catalina? Now, one of the things that I noticed in the paper was that they actually looked at pollutant levels, PCBs, etc., to see if there was something specific to the island, pollutant-wise, that might be responsible for this increase in tumours. The results from that were negative. They didn't find anything. There was no higher level of pollution on that island that could account for this increase in the ear tumours. But what does that leave us with? And just saying ear mites cause cancer is so simplistic, it's unreal. Because you have to ask yourself, why on earth would ear mites cause cancer? Why? There's no sign of a carcinogenic type of virus. I mean, we do know that wasn't that the problem with the Tasmanian devils and the facial tumours? It was a virus which was spread by bites.
I believe that was a virus, yeah.
Yeah, but there's no sign of that with the foxes so far. So what is it about this one population?
Can I ask a question? Why are they treating foxes?
It's because this is an endangered population of a very specific type of fox. It's endangered.
Oh, okay.
And they didn't set out to treat the foxes. I mean, it had been noticed that on this island, there was an awful lot of foxes scarpering around the place with these tumours on their ears.
A little over 50%.
Yeah. But on the other islands, you didn't see that. Why? What was it about this one population that meant that they had these tumours and the other ones didn't? So that was the observation on which the research was then started. It wasn't a case of what's causing the tumours in these foxes and, leaving everything else out. It was the fact that there was such a difference between the islands. This is odd. This is a very odd observation. What's behind it? Now, if we look at, causes of cancer that we know of, that are well established, constant irritation is one. Everybody knows of the person who had that denture or tooth that rubbed for years and they ended up with cancer in their mouth. Anywhere where there's chronic irritation. So there's a constant healing process going on. Healing meaning cell division is upregulated. So there's more cell division. Any process that requires that, increased cell division, means that there's increased chance of an error in the DNA when division happens. More divisions, increased risk. Therefore, increased irritation, increased divisions, increased risk. So we know these mites do actually cause some really nasty irritation to the fox's ears. So we've got increased irritation. But we've also got increased irritation in the ears of the foxes on the other islands. So it can't just be down to the mites. What could it be? So the next question that anybody would ask is there anything genetically different about this population of foxes? And when we go back to something we mentioned earlier, the population crashed because of the canine distemper outbreak, and the populations have recovered from a very small number. And we know that that type of bottleneck reduces genetic diversity. And we also know of a thing called the founder effect. You want to explain that one, CRISPR?
Yeah, sure. I mean, so about 30 to 40 years ago, there were maybe 2,000 animals on the island, and the canine distemper outbreak had something like a 95% mortality. It was just extremely bad. I left with maybe 100 animals or so on the island, and that type of bottleneck, there's a tendency that just random chance you'll have the survivors will not be a fair representation of the prior distribution of alleles. And so, just by chance, instead of having 1 out of 10 animals with a certain trait, you'll have 1 out of 2 animals with that trait. And once you have that huge initial increase, it becomes very easy for that allele to move to fixation in the population because the population is so small. And so it seems likely that's likely what happened, is that there was some pre-existing genetic predisposition for this, but you'd only really have animals that were heterozygous for this trait, and not that many. But then you lose 95% of the population by chance. There's a greater proportion of the survivors that carry this, and then suddenly, after a few years, the population's effectively all homozygous for this trait. And then suddenly you start seeing the tumors.
Yeah. And one of the things that we do know can cause that sort of predisposition to tumor formation is some sort of fault with DNA repair mechanisms. We see that in humans, that if there are faulty DNA repair mechanisms, people are more prone to developing cancers. I remember watching a documentary about an entire family who had, it was a genetic, I can't even remember what defect it was, but they were all very, very prone to cancer, and especially skin cancer. just because they had a problem with one of the genes that looked after DNA. So you can see a scenario where a problem with one of these genes, which fixes any errors in DNA, if that was in the fox population and their ears have been constantly irritated and cell division is increased and there's a higher chance of an error during DNA replication, but these foxes wouldn't have the normal chance of fixing that error, then what's going to happen eventually is mutations will happen in genes that control cell growth, and the cells will then start to grow out of control. That's what happens.
Yeah, and it's important to remember that, you know, they're seeing this rate of 52% incidence, 52% incidence in 4-year-old animals. The lifespan of a fox is 5 years in the wild, right? Now, maybe on these other islands with the ear mites, if you had an average lifespan for the animals of 10 years, you'd start seeing the tumors too, but you know, they don't live that long.
Yeah.
So this is just, you know, and this is a very common idea in terms of tumor progression, is that there's really not much of a reproductive hit when you're going to be developing a tumor very late in life, because if you're going to have kids, you've already done it statistically. And so, and again, I think that's another thing that possibly some people don't understand. I don't know.
Yeah, because this is something that I've always hammered a big drum about. Evolution isn't about how many offspring you're able to leave behind. It's about how many grand offspring you leave behind. Your offspring have to be fertile and have to survive to reproductive age. So it's actually the grandchildren that you leave behind rather than the children. That's what counts. But I think a lot of people miss that one out. There are conditions in humans that don't manifest themselves until well after reproductive age. And there's a couple of autosomal dominant ones that are horrible. One is Huntington's chorea. And the other one is fatal familial insomnia, which is actually a prion disease. But that seems to be inherited as autosomal dominant. So you only need one faulty copy and you will get the disease. Why haven't they been wiped out in the population? Because the disease doesn't manifest until well after reproductive age. You've already passed it on. Anything that pops up very early in life is only going to stay in the population if it's a recessive allele that causes a problem, not a dominant one. Otherwise, all your carriers would die before they reproduced and the gene would be lost. So I think with these foxes, I think what they've done is they've actually got tissue samples from all of the foxes for further genetic analysis of the individuals.
For histology, not.
I'm sure that some of those samples that they've taken they're going to be using to look at the molecular biology of the foxes.
I would expect so, but I mean, specifically for these articles, they're mostly talking about the histology done. So I do believe that they did some staining for a couple of proteins that are associated with various things. I think that maybe Ki67, for instance, but yeah, I mean, in terms of, you know, really getting into the finer details of the genetics or anything, I think that that's planned for the later work.
Because this work actually, it raises more questions than it answers. It's a fascinating piece of work. And but the big question is, why this island? Why this one island and not the others where the ear mites are present? And I think one of the things that's getting lost in translation as well is when we talk about, well, we treat the foxes for the ear mite problem and the incidence of tumours goes down. It's the incidence within the population, not within the individual. Curing the ear mites doesn't cure the cancer in the individual.
Yeah, and that's something I had to specifically explain several times in the thread. You know, if you actually go through the data table, which I did because I had nothing else to do, I guess, for a little bit. You can see that there were some animals in both groups that were initially the initial biopsy found a tumor, no staging information. I should stress that very carefully. No staging information, but they did find a tumor. And then the final biopsy did not show signs of being a tumor. Did not appear carcinogenic. But this was in both groups. Okay. And no statistical significance between the rates of remission, if it is indeed remission, between the treatment group and the control group. Now, Tommy gets caught up with this this comment made in one of the popular articles that there's an apparent reduction in tumor incidence, referring to within a single animal, there's an apparent reduction in the tumor. But that word apparent there is quite important because what they're saying is you've got this ear canal of these animals, right? That's just loaded down with wax and debris and just, I don't know if anybody in the room has ever seen the inside of an animal's ear when it has ear mites, but it is not pretty.
No.
And of course, if you have clusters of ear mites, and they do like to cluster once there's a decent number, you're going to tend to get a little bit more localized inflammation wherever the clusters are clustering, which will give you a little nodule, right? A little bump. Now that's not a tumor though. It's just an extra localized swelling.
Yeah.
And then you'll get also a lot of extra debris in that area. And if you're not cleaning it out and carefully examining it, it looks like it might be a tumor, especially if you're looking at a population of animals that get tumors. But then once you clear them all these mites out, if that's not actually a tumor, if it's not actually a carcinogenic or a cancerous growth, if it's just localized inflammation, then it clears up because it's not actually a tumor in the first place. But yeah, there were a few cases where there were some likely benign tumors that did clear up, but you saw that in both groups.
And also, they did not biopsy every single tumor in every single animal. They biopsied a sample of them and did the histology to confirm that these growths were in fact neoplastic, i.e. cancerous. You have to have a look at the cellular level to tell if something is malignant or not. And once they've got the results, we're looking at these things, and yes, these are cancerous lumps. The only thing they can say is the ones he did the histology on, they are definitely cancerous. But when they come across the lumps in the other ones, which they have not biopsied, which they haven't done the histology on, they cannot say that is cancerous. They can say that's apparently a tumour, but they don't have the evidence to say it is a tumour. So they call it an apparent tumour, one which macroscopically looks like a tumour, but they do not have the histological evidence to say that it is a tumour. And that's why the word apparent is so important in there. And the big drum, yet again, I keep banging, is that when a paper is written up, every single word in there is there for a reason. You don't get to use superfluous filler words, because when you submit your paper full of superfluous filler words, the editors will tear you a new one. In the days of mainly paper journals, space was at a premium. You had to get your article or your paper that you were writing short enough. You had to be really, really stingy in the number of words you used. So you had to take out any filler words. You don't add an apparent for no reason.
Well, I have to say it's actually slightly worse than that, because the quote apparent reduction in tumor incidence was not in the article. It was in the Popular Science thing.
Yeah.
Right. So if you actually look at the article, right, they say that among mature SCA foxes, 76.1 had GPL, gross proliferative lesions. And 70.6 of those GPL were classified as tumors upon histopathological analysis.
Sorry, say that bit again.
Okay, so among mature SCA foxes, 76.1% had GPL, and 70.6 of those GPL were classified as tumors upon histopathological analysis. So in other words, what they're saying is 3/4 of the animals had things that looked like tumors.
Yes.
Right? But of those things that looked like tumors, only about 3/4 actually were. And so that's, if I'm reading this correctly, that is, well, it's actually, so 30% of the 75% not actually tumors.
Yeah. So that is why when they are looking at those ones that may look like a tumour, but they haven't done the histopath on, they will call them apparent tumours. Apparent meaning having the appearance of.
But they'll only even say that when they're talking to, popular level science writers, because that's a degree of non-specificity that just does not fly.
Yeah, and you cannot make that declaration in a paper. The number of cancerous tumours, unless you have the evidence to back up the fact that they are cancerous. So, i.e., every tumour you're talking about, you have to have done the histopath on, because otherwise it's sloppy.
Well, I say lesion there, not tumor. So they're not even looking at whether or not it's cancerous necessarily, but whether or not it's actually a tumor, right, versus a lesion. That's the distinction that they're making. And actually, so the sentence after the one that I read is although GPL were detected in 20% of SCI foxes and 32% of SNI foxes, none of these lesions were classified as tumors based on histological exam. So that means that these animals on the other islands that have the ear mites, they get these tumor-looking lesions, but they're not actually tumors. Not even that they're not actually cancerous. They're not even actually tumors.
I am attempting, hold on, I'm just trying to find the original article to post it into the live chat. One of your links isn't working, by the way.
So what was the point for Tommy posting this article?
Ear mites cause cancer.
Is he trying to say that ear mites are like the environment? Or I don't understand what analogy he's trying to draw.
No, just the ear mites cause cancer. So if you deal with the ear mites, you get rid of the cancer. And he's missed the fact that that's prevention and not cure. Seriously,
okay.
that's it. I'm just
not relating this back to humans and cancer at all. I find that hard to believe.
Well, he is in a way in that he's saying, see, I told you. There's the cause. Here's the treatment. What he's missing is that the treatment reduces the incidence within the population. It does bugger all for those that have already got the tumours. It doesn't cure the tumours. And he's mixing up prevention with cure.
Yeah, I mean, you know, it's sort of like if he said that, you know, saw somebody who lost a limb in a car accident, and he said, "Oh, well, just don't get into any more car accidents. The thing will grow back."
Mm-hmm.
Or put on your seatbelt.
Yeah.
Yeah, start wearing your seatbelt. Your limb will grow back. That's Tommy level logic.
Yeah. So it's. I found it at last.
Oh, and there should be an extra 1 on the end of that one link.
That would explain it, right? Because I found the actual link itself, and I will put it into the live chat for peeps to see. Okay. This is the original study on which the Popular Science article was based. Right. We'll see if get somebody to try this link once it's in the live chat, just to make sure that it's actually working. Right. Somebody, please click on that and see if you can actually get to that article. So that is the original article.
Works.
Sorry?
Working just fine.
Excellent. So that's the original article. And it's one of the very interesting bits is down within the discussion itself, which asks the question that immediately leaps out at me. Where is it? Come on, get into the discussion now. Here we go. Right.
Well, I definitely would like to add that things like mites or parasites, those things definitely will increase your risk for cancer.
Mm.
Those things would definitely increase your risk for cancer, but that's obviously not what Tommy was saying. He was saying that you can cure the cancer by treating the risk factor, which that's completely false.
Yeah. Yeah, it's to do with predisposition too.
Mm-hmm.
Now, I am going to attempt to share my application window. Let's see if this paper will come up, because I want to read out the bit that jumps out at me. And this is the crux of the whole article, which seems to have gone whee over the head, plop onto the wall. And it's just right here. Oh, God. My mouse is misbehaving. There we go. A key question is, why do ceruminous gland tumours only occur in the Santa Catalina foxes and not in foxes on the other 2 islands where ear mites are also common? What is so particular about this bunch of foxes? That's what leaps out at you from all of this data. There's a difference somewhere. Taking this data and saying ear mites cause cancer. Well, then how do you explain the other 2 islands where there's oodles and oodles of ear mites and no cancer? If the ear mites are causing it, why don't they have it? And one of the suggestions that was made was, well, okay, in talking about this, what we should do is, you're going to love it, ignore the data from the islands where there are no tumours and just look at the island where the tumours are. What? Ignore the observations that your idea cannot explain. That's called cherry picking, is it not? And what would happen if we did it the other way? Let's ignore the island where the tumours occur and look at the two islands where you've got oodles of ear mites and no tumours. Would we then conclude, oh, ear mites don't have anything to do with the tumours? No. And because it's looking at your population on this one island where the tumours are popping up, and yet with everything being the same, these tumours aren't popping up on the other two islands. Why not? What is it that's causing this difference? And it can't be the ear mites, because the ear mites are on all three islands. Again, I'm labouring the point here. It may seem really simple to everybody out there. And I'd ask in the live chat for people to let me know, do they get what I'm saying here? That it can't just be the ear mites, because if it was, all three islands would have the same incidence of tumours. Or am I missing something really bloody basic here?
Well, I just wanted to say really quickly, it's slightly worse than that now, because since they've been treating the animals on the one island for ear mites, there's actually now a significantly higher incidence of ear mites on the other two islands than Catalina, and yet you still only get tumors on Catalina.
Yeah.
I was gonna. I didn't. If you don't mind, I had only sort of glanced at that thread. I'm kind of to the point where it's like, oh, good, Tommy's pontificating on whatever, and it's, I'm always curious. If I have the time, I'll go through it. If I don't have the time, I don't really lie awake at night, worrying about what I'm missing. My recollection from that thread about what I imagine Tommy's point to be revolves around his rather peculiar use of the word random. So I think what he was saying, it was something like, okay, so you have these. Well, first off, I have to say, yes, it's a tremendous case of cherry picking, or as I often refer to Tommy's, needle in a haystack research. If you have an island that has foxes with ear mites and tumors, and you have an island with foxes with ear mites but no tumors, he can ignore the one island and pick the one that best suits his cause. I mean, that's as you pointed out, Fiona, with the support of other people in the room here, that's very much not how you proceed in figuring out what causes what. But that's, I think, the point's amply made by you guys. But so let's just focus on the one island. So you've got some foxes, you've got ear mites, and you've got some tumors. I think Tommy's position is that tumors are something that are designed, that is to say, have the intelligently intended purpose to do a particular function. And this function arises on a as needed basis, right? So when you have too many toxins in your body, well, then your tumor system will activate and jump into action and help take care of those toxins. In the case of the ear mite, ear mites, well, there's some problem that the ear mites are causing, and then your tumor whatever system activates and jumps into. So the one line that I had seen of his where I went, okay, yeah, more Tommyism, and I just sort of scrolled past and didn't read the whole thing, was when he said, see there, in a kind of way like, oh, this blows your whole, this blows your whole worldview out of the water, was there, see, tumors don't happen randomly. You people all believe that tumors just happen randomly. And that's crazy, because when you look, you see these tumors happen when there are ear mites. So there.
Yeah.
And this is kind of something Tommy's been on for a while. And I think where we sometimes struggle with what he's saying is because the thing he's saying is such a dog bites man story, right? Nobody thinks that tumors happen randomly in the sense of like lightning strikes or popcorn kernels popping, right? Like where you just, I don't know, there's a tumor, there's another tumor. We don't know why that happens, right? We understand that there are several correlates.
Mm-hmm.
That in the aggregate, when you analyze the data, some of these things are causative roles. So, for example, there's a genetic predisposition, and then there's environmental factors. And everybody knows the story of somebody who smoked 2 packs a day until he was 120 years old and never had a tumor. And everybody knows somebody who was a marathon runner and lived super healthy and had a tumor when he was 35 years old. And why does that happen? But for the most part, there's some very reliable correlations there to where you can infer reasonable causative roles, so that there's a predisposition that's established genetically. There's environmental factors that tend to instantiate or potentiate the stages of tumor development and right up to metastases. And there's nobody out there arguing that tumors happen just randomly. They just fall from the sky and we don't know why. And of course, there's nobody should be arguing that they happen as a precise response to particular environmental factors. Let me draw as a contrast the platelet aggregation and so these activities that happen around when you get a cut on your hand, right? There's a very reliable thing. You have these what do you call them, gobbies? What's the word used for it? There's a word you use for it, Fiona.
A word to use for what?
Gobbies. We talk about little
gobbins.
Gobbins. Gobbins. Yeah, gobbins.
Yeah, so you get aggregation, so you get clot formation, right? This doesn't just happen all the time. When you cut your skin, it's a system that sort of jumps into action to reduce the blood loss and to begin the process of healing. And this isn't something that just happens randomly. It happens in response to a specific environmental triggering, and it happens in everybody all the time, unless, of course, there's some pathological state.
If it's happening randomly, you're screwed.
Yeah. Well, that's the thing. There are actually two different pathological states, like any other system, physiological system, and that there's some people where that system is tuned too slow and it doesn't react quickly enough. And there's people that it's tuned too quickly, and then they start having clotting all over, right? But for the most part, the vast core of humanity, you get a cut, then this system jumps in. You can say, well, that system is something that is reactive to getting a cut. So, I mean, nobody should make the claim that tumors just happen randomly. We don't know why. And nobody should make the claim that tumors are a programmed, precise response to specific environmental stimuli. And nobody is making either of those claims unless you don't know what the fuck you're talking about. There's my thought.
Yeah. I said in the thread somewhere that everything that Tommy has ever said about the conventional view on tumor progression is somewhere between a straw man and a red herring.
Yes.
Yeah, exactly. And also it gives them that's a great point. It's a great way to put it. And it gives them the maneuvering room too, right? So that when we say, like, you're pointing this stuff out now about, well, there's this other island, by the way, Tommy, you didn't mention this other island where they have these tumors. So there clearly is it's not the case that these mites cause the cancer, right? There's obviously a significant genetic component. I hear you guys talking about the bottleneck effect. And now Tommy can say, well, I never said that genetics weren't important. Oh, you guys, right? That's what you're going to get.
Yeah. Oh, listen, we've already had them ask, given that the incidence of tumours within the population dropped off when they started treating these ear mite infections, Tommy has demanded that we explain how treating the ear mites caused the genetic change in the foxes.
Yeah, when I first saw that from him, I initially typed, "Is that a joke?" And I got rid of that, and I asked him if possibly what he was trying to say is that due to the long-standing chronic irritation caused by the ear mites, you get an increased rate of mutation in the cells of that local tissue, right? Which, in addition to the underlying genetic predisposition, leads to cancer. In which case, you've caught up with us 300 years ago. Congratulations.
Yeah, because when I saw that comment, and I thought, nobody has suggested that treating ear mites changes the genetics of foxes. That's not what anybody said anywhere. But it was one of those comments that when I read it, it was a sort of, I got nothing moments. Now, here's the other thing that's been baffling me. Jesper, you put a comment in the side chat. Instead, what is happening is that the tumour is indeed a protective mechanism generated by the body and put in place to seal off infection, dead cells, or lactic acid from the rest of the body. So how come all these foxes that have put this protective mechanism in place are all dying from cancerous tumours? Some protective mechanism. The mites aren't going to kill them.
Yeah, and to be perfectly fair too. I mean, I know that there is a decent incidence of mortality due to these tumors.
Yeah.
But if they're mostly occurring in animals that are over 4 years old and the life expectancy of a wild fox is 5 years,
Hmm.
I mean, yeah, sure, that's the thing that kills them instead of something else, but they weren't going to make it much longer anyway.
Hmm.
Still bad, especially with
Yeah.
such an endangered species. You want to do the best you can, but I mean, it's
I am
I,
trying
yeah.
to find the causes of death in the foxes that they found. It was things that were a result of the actual tumours themselves, with septicemia, overwhelming infections, etc. Because, of course, some of these lesions are fungating in the later stages. I'm trying to find it.
Not to mention, I'm
I've
sure the
got it.
foxes itch those tumors.
Hmm.
I've got it here. Some carcinomas were the proximate cause of death due to metastasis or sepsis secondary to tumor erosion of tissue and abscessation. Other tumors, both carcinomas and adenomas, also likely contributed to morbidity in some animals due to secondary bacterial and yeast infection.
Yeah.
Because adenomas appear to be on the same developmental pathway as carcinomas, and some foxes diagnosed with adenomas later developed carcinomas in the same year, we also consider the high prevalence of adenomas to be significant to this population. So yeah, there's definitely some of these animals that died because of these tumors,
Yeah.
either directly or indirectly.
Yeah. How many incidences of death have there been from ear mites alone?
Well, you must have had at least a few cases of a fox scratching a little too hard and getting an infection in the cut that it opens up.
Yeah.
That's the best I can do.
So these tumours that seem to be able to kill off the foxes are the defence mechanism against the ear mites that don't kill them. Has anybody else seen a problem with that? Or is it just me?
And of course, given that that's the only thing I can come up with where the ear mites would even indirectly lead to death, I'm guessing that having an actual cancerous tumor growing in your ear would itch even more.
Yeah.
Yeah.
So as I say, it's not really the protective mechanism that I would really want happening if it was me. It's like it's like curing the pain of the ingrown toenail by putting a bullet in your brain. Your ingrown toenail no longer hurts.
Well,
But
not only that, Fiona, but it's not good for the mites either that the fox dies.
well, there is that
The
too.
lose. Nobody wins.
Yeah. Now, I'm just looking at the paper itself. They did look. Somebody's asked in the live chat. Did they look for fungal, bacterial, viral agents being involved? Yes, they did.
Yes, they absolutely did.
They actually did. They also included, and I'm trying to pull it up now. They also looked at pollutants to see if there was a higher amount of, I think it was polychlorinated biphenyls.
Yeah, I've got the line here. Yeah, they did test for that, and also chlorinated compounds. So we detected low levels of PCBs in the sera of 2 foxes. PCB and OC levels in adipose tissues were highest in SCI foxes, which is not the island we're interested in, with 4 of 5 having quantifiable levels. Yeah, so the lowest levels were in the SCA foxes, and those are the ones that have the tumors. So go figure.
Maybe the ones on that island, the ones with the tumors. You guys are looking for why can they get ear mites on these 2 different islands, but then only the ones. Are the foxes on the island with the tumors, are they drinking tap water? Is that the thing?
Vitamin B17, Hogtie.
Well, I mean, it's just,
Yeah.
yeah,
Their deficiency. Yeah, there's not enough B17 on that island.
yeah, I mean, they literally found that the lowest level of Tommy's favorite toxins here were in the animals that got all the tumors.
Oh, yeah, okay, yeah.
Yeah.
Sorry, I was away from keyboard, but I overheard part of this, and I actually, strangely, I know the answer to this. I've taken a lot of entomology classes, both agricultural, did I say entomology, agricultural, medical entomology, and of course wildlife entomology. And it turns out mites are not really dangerous to any of the canid populations, except there are they do get to be dangerous to the population when they carry some other organism.
Mm-hmm.
Right, so I mean, you're going to know stories like was it ticks? What causes Western equine encephalitis?
Yes.
Like horses that live in the wild have all these bugs on them, and they really don't care. They don't really bug them. But then they can get these other little critters that live in the mites that then affect that horse population, and that Western equine encephalitis is a terrible thing. I may be more familiar to the mostly humans listening to the broadcast. We don't get a lot of horse followers, and Lyme disease, right? So I mean, you can get ticks. We have ticks where I live, and nobody cares, except that then now we get Lyme disease. That's
carrying Borrelia burgdorferi.
Yeah,
unless it changes its name.
Yeah, there's a Sarcoptean scabies. Okay, people know what scabies is. Yes. So there's a wild canid version of scabies that comes from one of the Sarcoptes mites.
Mm-hmm.
And that's really unpleasant. They're burrowing mites, and it's a really unpleasant thing. The canids will actually like scrape off sections of hair, and it's a really stressful thing for them.
Yeah.
And I don't ever. I mean, why don't I should search for this before I open my mouth. But my recollection has been there's no mention of like, oh, it'll depopulate all these foxes or coyotes, for example. Coyotes can get scabies too.
Yeah.
Sarcoptean version that attacks the western coyote populations, both in the southwest in the States and northwestern Canada. But they don't. It's not a fatal thing, right? I mean, it's stressful for them, but nobody dies from it.
And thank you, because I mean that reminds me of something else. He kept going on about how you would expect this big die-off, and how could you have a difference if you didn't have a die-off and then replacement with new animals, and like.
Sorry, a die-off from what?
Well, he's thinking that the tumors are going to kill all these animals. It was like no, that's not how epidemiology works.
No, sorry,
they're all going to die at once.
Yeah, there's two things. Again, I didn't follow this, and I didn't do any. Like I was out when I saw the message for this hangout, and I sort of came back in. It sounds to me like I'd come back in about 5 minutes after it started, but so I didn't really look into this stuff. Does anybody know the species of mites that affect these particular? Because I don't know these foxes. I don't know where the hell we are. Are they Sarcoptean or something else? Or
hold on, I'm just looking them up.
I think Hogtie's making up words now.
What's that?
Just kidding.
If you ever take entomology, a lot of memorization in entomology. Sarcoptes scabii is the name that I remember.
It's
IEI, something like that at the end.
Otodectus cynatus, maybe.
I don't know what that is. Well, now I'm stymied. I don't have no clue what that is.
It sounds like it's Latin for ear mites.
What's the science, bitches? Boy, that's. But sorry, the other question was the death rate to the tumors, right? I mean, generally the tumors, even in people that are sort of on the surface, are not all that quickly fatal. I mean, any tumor left rampant will. I mean, a lot of people get tumors, even something like colorectal cancers and some of these masses that form. I mean, you can carry them for a decade and get hit by a bus. Prostate cancer famously is the one that a lot of men die with, but not from prostate cancers. So, I mean, I don't know. I mean, why am I calling in? I'm not an expert in fox ear mite tumor mortality rates, right? Like, I mean, did anybody look into it? Do they actually die from the tumors? I wouldn't think they would.
These tumors are then going to allow in opportunistic pathogens. They're in the ear, so they are close to the skull,
and of course,
structures, rather crucial structures.
They get Yeah, I mean, being in the ear, they also have really easy access to the lungs, obviously, too, right?
Yeah.
So you've got, it's not really good. The opportunity for really fatal metastases is pretty high, but
Well, sorry,
yeah.
where are the tumors then? Are they? I thought they were in the, like, foxes famously, all the canids have external pinna, the triangular shaped.
It's
yeah,
in
so
the wax glands.
it absolutely anywhere in the ear canal.
Yeah.
No, sorry, where the tumors are? They're
Uh-huh.
not. They're
Yes.
not on the pinna. Oh, okay. Well, see there. Now I learned something. Good science, bitches.
The,
No, I
basic-
thought you were
the
talking
affair
about.
the parasites are found.
Yeah, and
okay.
have you ever seen the inside of an animal's ear with ear mites? They're
No,
not.
never.
They're not on the
No.
external bit.
Yeah. No, I've seen like the coyotes. I mean, we have, like, say, we have, like, well, not the ear mites. No, anyway, just answered. No, I assume that it was on the pinna. It's down in the canal.
It's the ceruminous gland tumors, both carcinomas and adenomas.
If you see the link that I've got on the outside, you can. And actually, if it's my link, you're gonna have to add a 1 to the end of the pathology one. But yeah, it's got some images there, including a metastasis that they pulled out of a lung.
Mm-hmm.
But yeah, it's
Let's
See,
put it this way, the tumors are worse than the mites.
Do you mean the live chat or the Hangouts IM thing?
Are you looking for the original article again?
let me just repost it on the inside.
Yeah, just so I can. Yeah, I'll take a look. I mean, now that you're telling me about the location. Oh, now I'm hearing myself. Oh, that's me. I did it to myself. JD came to myself here. Yeah, no, I mean, I'll take a look at the images, but I mean, there's no like, I mean, if you get tumors, yeah, if you're talking about now glandular tumors, like, yeah, that's bad.
Yeah, I mean, there
I'll
were
give
also
you that,
ear
but I gotta
lesions
look at
as well, but the ceruminous gland tumors were detected too, and identified in a sample group using histopathology. So they were shown to be actually malignant. So you've got this incidence of cancerous tumor formation in the ears that is higher on this one island. They've confirmed that it is cancerous. It's not just a case of, well, we saw some lumps.
And I got to say on the histology, I'm certainly no expert on this, but you look at those images, yeah, that's bad. Like,
Yeah,
I wish
sort of jumps out at you.
I wish these images came with some sense of scale. Oh, actually, there's tools in this one. Oh, that's big. Yeah, no, that's big. That'll kill a fox. Okay, I see what you're saying now.
yeah. I like the way that's big. That'll kill a fox.
What imagery are you looking at?
Yeah, I mean, there's one of them. Figure 3. It has invaded into the adjacent bulla, tympanica, pharynx, temporal and occipital bones, and the skull of the fox.
Yeah, that's unquestionably fatal. I mean, it's a matter of time, right? I mean, they're not. That's not a fox that's waiting around to get hit by a bus. I mean, that's unquestionably fatal.
Yeah. So I mean, these tumors are aggressive, and I mean, but here's the toss-up. Some might that is a protective mechanism.
Oh, I'm sorry. I'm well past that. I'm just trying to catch up with what you guys were talking about. No, I mean, clearly it's not a protective mechanism against mites. Like, again, I feel speaking a little bit out of school because I haven't studied this particular mite, but there's no famously fatal mites amongst canids. As I say, there are some that carry other diseases, but no mention of that here.
Well, there is an increase in the malignancies in the ears of domestic dogs and cats with huge ear mite infestations.
Of course, though, again, remember that domestic lifespan is
Exactly.
hugely increased over
Yes.
wild lifespan.
But the one other thing I wanted to say was, Hogtie was mentioning, if these mites are carrying something else, and he mentioned West Nile. Was it West Nile virus? Oh,
No, I was using it as an example, and that's not a mite.
Yeah.
It's its first cousin, the ticks, the Western equine. I'm going to look it up just to make sure I haven't got the wrong one. It was, of course, it was a few
This
years
West
ago.
Nile virus.
Western equine encephalitis.
Yeah. But they also looked to see if there was any herpes viruses or papilloma viruses.
Uh-huh.
To see if there was anything like that. These viruses are known to increase the chances of cancer. Because if you remember, there's that horrendous outbreak of tumours in Tasmanian devils. And it's been found that that is caused by a virus in the saliva. And see, as Tasmanian devils fight all the time and bite each other. And they tend to bite each other on the head and the face and the neck. They were getting these tumours at the bite sites because the virus was being injected in with the bites. So they've had this massive epidemic in Tasmanian devils, but they managed to narrow that down to a virus that was carried in saliva. So with these mites, they had a look for those viruses known to cause cell proliferation. These viruses have been associated with tumours, and they didn't find them. They didn't find any of that at all.
And let me just say, don't do a Google image search for Tasmanian devil cancer if you've got a weak stomach.
No, don't.
Yeah.
That is, although having said that, there's a certain fascination. But don't do it, guys. Take CRISPR. Don't do it. Just don't look. I did it once and I had nightmares.
Nope. Special update, science bitches correction.
Well,
no, it's not any of the 8-legged buggers. They're dipteran, two genera of mosquitoes. So they are actually flies that transmit Western equine encephalitis.
Mm.
On this page they call it encephalomyelitis. Well, why wouldn't they call it that? That's what it does. Yeah. Well, that test was more than a few years ago, I got to admit. So it's mosquitoes, isn't it? Oh, well.
Now, here's my next question. What treatment did they give these foxes?
Oh, come on. That was going to be my closer. You stepped on it.
I'm sorry, but we've been going for quite some time here.
Yeah, that's a good point. So this is my favorite thing from all of this is that here's Tommy going on for weeks and weeks and weeks about how toxins, got to get all those toxins out of your body. Toxins, they cause cancer. That's all they do. And except for foxes, let's flood their ears with lots of toxins. That'll cure cancer. Where'd that come from?
Yeah, I mean, do you know what it was they were using?
Well, don't say DDT. That would be too fun. Nobody uses DDT.
Ivermectin.
Ivermectin.
Good choice.
Ooh. Yeah, ivermectin. Ow.
I haven't seen him mention that one. Is he had any like, what am I trying to say? Has he specifically said anything about that being carcinogenic? I've never even heard anyone.
Well, I got to say,
said that.
Given the choice between that and Roundup, I'd rather the Roundup. I mean,
yeah.
What do you guys have against ivermectin?
Yeah, I'm just having a look at
what do I have against it?
Yeah,
nothing. If it's not being administered orally, right? Like, I mean, that's Tommy's big thing, right? It's that you somehow getting huge amounts of these toxins into your body somehow. Like, yeah, I mean, ivermectin on pets as an anti-parasitic is great, but just like Roundup is a great way to kill some weeds. It's not, and again, that's less toxic than ivermectin. I don't think you would disagree with that, right? I mean, because I
don't know how toxic ivermectin is.
Yeah, I'm catching up.
Decently.
I'm catching up on it. What is it? Like, how does it work? I mean, it looks like it kills lots of arthropods. Like it's
it's a lot higher in mammals than it is in arthropods, but it decently readily measurable, though. I mean, it's, you have to drink a couple of gallons of Roundup for the active ingredient to kill you. So,
I mean, it's a multi-ring, multi-cyclic, interspersed. I mean, it's a big structure, but then I don't see anything. Oh, it kills this, it kills that, it kills this, it kills that. How does it kill things?
That's what I'm looking up right now.
Central nervous system depression in mammals and consequent ataxia. Nobody wants that.
Yeah.
Well, I mean, yeah, that's actually so. It works the same in mammals as it does in arthropods, I think, but the specificity is much higher. So it does actually block the transmission of the nerves in invertebrates, but with a very high degree of specificity, if I'm reading this right. So go ahead.
I'm only up to the part about it has a GABAergic effect. Worded on
it as well.
Potentiation of inhibitory GABAergic synapses.
Yes.
So, I mean, what, it makes you sleepy? It makes you, I mean, that the worst things can happen. I just don't see what the mechanism is. You're saying it actually reduces nerve function, and yet it's GABAergic, meaning it increases translate. It increases activity in a system in mammalian brains that tends to inhibit other activity elsewhere.
Yeah. In other words, it's nasty stuff.
Right. I mean, okay, so I'm just seeing LD50 acute in mice is 25 milligram per kilogram. So that's decent, right? I mean, it's pretty. That's decently toxic, right?
Yeah, I found it.
Here's what I did. It's actually glutamate channel chloride ion.
Yeah, okay. Yeah, you buzzed in first. That's fine. That's fair.
But it's specific for invertebrates in their nerve and muscle cells that produces paralysis and eventually death. I don't know that it's actually very toxic for people because we give pretty big doses.
Well, I mean, it is. It's 25 milligram per kilogram in mice, so it can't be that far off in humans.
Yeah.
Well, because I would say like 25 milligrams would be like the dose you would give a person who's like more than 70 kilograms, like 80 kilograms. You'd give them like 25 milligrams.
I got a lot of kilograms on me. I'm feeling like I could take a lot of this stuff.
It doesn't cross the blood-brain barrier either.
Whatever knocks back the scabies, man, I'm all for it. Coat me in
stuff. But the scabies obviously is a lotion. You don't take anything orally.
Yeah, and again, I mean, it's much more toxic to the invertebrates than to mammals. That's the important thing, right? Is that the dose that you need to kill off all of the invertebrates is quite low compared to what would be a highly toxic dose to the mammals you're administering it to.
Yeah,
but it is still, if you've got the box there that you're going to be administering it from, the box has enough to kill to kill the animal if you give it all at once. That's why you don't do that. I wish there was a sort of a more chemistry focused site that would give a little more of this information. Like there's no information here about permeability, membrane permeability. But did you look at the molecule in the corner? Like, I mean, anybody want to put some odds on how quickly that stuff passes through the skin? Like it looks like some kid went wild with Lego bricks. This molecule is huge. There's just no way this thing.
The LD50 in dogs is 80 milligrams per kilogram. But in dogs with an MDR1 gene defect, the LD50 is 0.2 milligrams per kilogram.
What administrative method?
Sorry?
How are you administering it for that?
I have no idea. This is just the acute toxicity of ivermectin.
Probably oral, right?
I think it is. Most of the ones I've been seeing, it's been oral.
Well, that's why I mentioned the molecule shape, because I mean, I'm kind of got the feeling you could get in a bathtub full of this stuff. Like it's the drinking it that's your problem.
I thought I saw on the skin. Hold on.
Well, here's a little bit more about its absorption. It's very well absorbed.
Here, it's
To get the percentage, but it says it does about 4 hours for peak concentrations in the blood.
dermal is, I think this is in rat. Dermal is 600 milligram per kilogram. So, bathtub full of it, probably a mistake.
Yeah.
Okay. Well, that wasn't medical advice.
Don't they
Nobody
use it in sheep dip?
Well, I mean, unless it's a bathtub full of it that's been diluted,
Yeah.
right? I mean, the original concentration, bathtub full, bad idea. Several drops in a bathtub, I think you're fine.
Okay, here's the general symptoms of ivermectin poisoning: ataxia, hypermetria, disorientation, hyperesthesia, tremor, mydriasis, recumbency, depression, blindness, and coma.
Sounds like neurologic symptoms to me.
Yeah, I mean, it says, Hogtie, pay attention. You're seeing as you want
Yeah,
to bathe
okay.
in it. Overdosing can occur due to excessive licking.
Yeah. Well, I mean,
after
we're
pour-on delivery.
yeah.
So it's the one you pour it on your cow, and if the cow starts licking it off, it can end up overdosed.
How do you stop the cow from licking on it?
Well, with cats, you put the wee drop of whatever it is on the very back of their neck. They can't lick there.
Uh-huh.
The problem is when one cat licks the other.
Oh yeah. You got to put cones on all of them.
Okay.
Yeah, we're kind of making light of it. Obviously, this stuff is poison, and I mean, people know.
Yeah, and, I mean, so the dermal LD50 is 24 times the oral, so
Yeah.
that doesn't help.
But basically, it's still something that's really toxic that you're putting there to kill the mites that does have an effect on mammalian tissue. And you're adding this toxin to your foxes, and the incidence of tumours in the population goes down.
Well,
not up.
Yeah, and that's, I
Well,
mean, we are making light. I think Fiona, you have a very familiar audience. People know, when I'm joking about getting in a bathtub full of this stuff. But I mean, the original question, why we all started rapid researching, this particular agent, was because we're saying, well, the toxins cause cancer, right? And yet, of all the things we're listing, they all appear to be neurological effects.
Yeah.
So it affects, it grandly affects the calcium chloride channels of invertebrates to where it's fatal for them.
Mhm.
And then you have these other effects, ataxia, and the GABAergic responses and things like that in mammals. Generally not fatal, but it's possible if you eat enough of the stuff or absorb enough of it through the skin. I mean, you can die from it, but nowhere. Oh, a person should probably do. Let's do a Tommy Hall search in here.
That's
Let's
what I'll
do.
do.
Here's a link for you guys.
Ivermectin causes seizures. Ooh, causes cancer.
That's what I was wondering about. That's what I was looking up at the moment.
But I mean, the reason why I raise that issue is that there's one of these things about, well, toxins cause cancer. I mean, we got a lot of toxins in the world that mess up your neuronal function, often to the point of death. But they're not necessarily. I mean, not all toxins cause cancer. There's a lot of bad toxins that doesn't matter what the dose is, you're not ever going to get cancer from it. Even sustained dose over the course of your life, then not all toxins are carcinogenic, and your body doesn't respond to the presence of these things by saying, "Oh, there's some toxins. I better." Build up some fat or make a tumor or whatever else, like in response to.
You're going to love this one. Ivermectin as an inhibitor of cancer stem-like cells. Medication that treats parasite infection also has anti-cancer effect.
I just saw that. Where is that? Science News.
Yeah.
That's fairly new.
2018 paper as an inhibitor of cancer stem-like cells. It's the NCBI.
Exhibits an anti-tumor effect, epithelial ovarian cancer, by interacting with KPNB1 gene. Huh. Well, that's fascinating.
It is, isn't it? It is. I'm just going to put this link in the outside chat. So, there we go. Linky in
I love how
outside chat.
your show has now become pro-toxin.
Has it? According to who?
Yeah, I don't know. I don't know. That's it. That's what'll come out of it. I mean, I think we're just trying to rebut the obvious sort of sophomoric silliness of saying, you know, well, cancer or toxins cause tumors, and you know, tumors are kind of an intelligent response. And tumors are not random; they're an intelligent response to the presence of toxins. And it's the truth is a lot more complicated and genuinely fascinating than that narrative.
Yeah, and this thing with, I mean, going back to the study of the foxes on these islands, this observation of the tumors only being on the one island, it's absolutely fascinating. I mean, Tommy's read the paper and looked at it as the answer to the question. All I'm seeing is a whole bunch of new fascinating questions. Why? Why this one island? Why not the other two islands? What's the difference? It's not in the mites.
And one of the really interesting things about it is, you know, here you have this opportunity where you do a genetic analysis of the different populations. What if the responsible gene is one we hadn't found before?
I know. That was my next bit I was going to be heading in the direction of, because you're going to have to look at these populations and try and find out where the differences are. Is this a difference in the fox populations that predisposes one population to these tumours forming? And looking at the data from the 3 islands with mites, that's what that data is telling you. There's something different about those foxes. Now, if you do as Tommy advises and just ignore the islands where the tumours don't occur, so all you do is look at the one island where the tumours occur, then what? What do you learn?
Boy, I'm with you. I'm like 20 seconds behind what you're saying.
Well, then you learn.
Sorry, it's just I just got to that part about the tumor prevalence and risk factors. You guys are reading the thing you're talking. It's the Vickers Clifford paper that you're talking about, right? Is that the one that started all this?
Is that
if that's been shared with the audience? I mean, you go down and take a look at the tumor prevalence and risk factor analysis, and there's a bunch of chi-squared analyses, blah blah blah.
You're also going to want to take a look at the Moriarty paper, which is the same group in the same year, and that is the one that actually talks about the treatment.
Well, I accept your endorsement, but I kind of don't want to look at it. I got to, like I say, I skipped supper because I just got supper thrown in front of the kids, and then this is interesting, though.
It's absolutely fascinating, because when you come across an observation like this, you've got a chance to discover something unique about that population. You may discover a new gene that plays a role in the formation of tumours, that when it is broken, it means that tumours form more readily.
Yeah.
Well, you don't know what you're going to find.
I don't see anything about the genetic. So what was the one? I mean, I was being dismissive there, but CRISPR was encouraging me to read something else. But where's the link for the other one?
It's also in the outside if you scroll up, or I'll just paste it again.
I'm sorry, I suck when I do that. But there's just so many chats that. Oh, is it? You've got a thing that says thread and then pop one. Is that one that's got?
And so in both of these articles are fairly quick reads, I think. I mean, there's nothing in it that's. This is not like a heavy mechanistic paper, right? I mean, you get through it pretty fast.
Yeah. Well, I tell you what, because I do see now in that, you know, the Hangouts, you know, the green bar, whatever thing called, you've got one post in there that's got several links to it. So I've got those, and those are preserved even when I close this other window. So it is interesting. But as I say, I do try to hold in balance. Like, I mean, do I want to get fascinated by fox ear tumors and Tommy Hall's take on science, or go watch a movie with my kids, right?
Well, I would say
I'm at that stage, right?
I would say keep it in the back of your mind and give this group a year or two yet to finish whatever study they're working on now, because what they're working on now should be the genetic analysis that answers the questions they raised in the first place. And that'll be the one that will really be interesting. So.
Yeah.
Well, you know what? Let me say this on exit then. I'm going to say it's too bad these foxes' ears aren't as exposed to oxygen as my testicles are.
Well, you see, there is that hogtie charm. I mean, listen, do you wear boxers or tighty whities?
I gotta say,
Hangout just took a weird turn.
Yeah,
you know,
it did.
I gotta say,
boxer brief, is that the right term for it?
What I don't
was
understand is, does he not have like skin over them? Because
They jingle.
because
You run.
I thought they were supposed to have skin over them,
Skin and fur.
that like protected it from direct access to the air.
Yeah, but no, I was just wondering what hogtie wore, because I thought, well, maybe we could put that in the fox's ears. All these wee foxes running about with tighty whities on their heads.
You just got to shave the fur off of them. Now, I'm not going to specify whether I was responding to Fiona's point or CRISPR's point, but shave the fur off of them.
Why not just wax hogtie?
Again, this hangout's taken an unpleasant turn.
It has, hasn't it? Yeah. And just this is for hogtie champ, right?
Yeah.
Hakuna Matata.
Kahuna Matata. I am still laughing about that. I find that so funny. If people know what we're talking about, there's you'll see it on my Google Plus feed. I posted a thing. Some Canadian creationist nutter wrote a thing to Aaron as a Microsoft Word document. The thing is hilarious as hell. He's taken snippets out of it and put it up on his Patheos page. If you're looking for it, it was posted several times in some of the discussion groups, notably on Great Debate Community. And I posted a thing about how the person had said that, you just don't have the kahunas to debate Kent Hovind. Really? Kahunas? Actually, and it's funny, I messaged Aaron yesterday about that when I said, like, did you not notice that the person put kahunas? He says, there was just, I didn't, he said, I kind of thought about making fun of that, but there were just so many other little jokes. I was going to let that one go. I said, that's okay, I'm on it. I'll put it up on Google Plus, the kahunas.
I must admit, when I saw your post, that just popped into my head, and I've no idea why. And I just had to put it out there, and I didn't realise it was going to affect you like that. Sorry.
Oh, that's awesome. Hey, yeah, I'm going to go check this out. And the kid's tooth's bleeding. I don't know why.
Oh, dear. You take care then.
Well, she's at that age, right?
Bleeding teeth.
Yeah.
That
Well,
age.
you know what? I'm a very good friend who's a dentist who has his practice like 2 blocks away. She just held up 6 fingers because you're being shy. You don't want to talk. But she had one front one that was loose, and they were goofing around in the truck, and she just they cracked heads, and like this thing just popped up like the hood of a VW Bug. And I was freaking out. I don't like teeth or fingernails. I have discomfort with it. So luckily my buddy, the dentist, was like, yeah, bring her on in here. And he used a device that he called tweezers, but it was actually a big pair of pliers. But he calls them tweezers in front of the kids. Yes. And boink, out they went. He's a pretty good guy, isn't he?
Well, but at first he put the numbing stuff on. After, headache.
He put the numbing stuff on, and then what?
And then he took the tooth out, and then headache.
You had a headache? Oh, that's right. Yeah, well, that sucks. Well, let's go check out the other one. I'm going to go take a look here. But hey, always fun seeing you guys. You know what it's like. Yeah, but
you take care.
See ya.
See ya. That means the Tooth Fairy's coming to Hogtie Champ's house. Think about it. The Tooth Fairy. You do all know about the Tooth Fairy, don't you?
Tell us about the Tooth Fairy.
You've never come across the Tooth Fairy?
Nope, I've never seen a Tooth Fairy.
She goes about a pair of a set of ladders and a pair of pliers. Because if she doesn't have the right change for the one tooth that you've left.
Hey, my Tooth Fairy never had that.
No, but I mean, if she doesn't have small enough change, what she has to do is pull another tooth out.
Okay, by the way, I haven't said anything because there wasn't really any need. The bottom link is to a cell.com paper about the genetic diversity in the island foxes. And the one link I posted above is an article from the New York Times about it. Let's take a look at this. This is it's called Genomic Flatlining in the Endangered Island Fox. Now this talks about all of the island foxes and highlights genomic monomorphism on a scale not seen previously in eukaryotes is shown. Deleterious variation and loss of function mutations are not purged. Drift without balancing selection accounts for peaks of heterozygosity. Persistent despite the absence of variation questions its role in small populations. And of course, there's a lot more. The link from the New York Times that I published, posted, sorry, has a video of these foxes. These are little tiny guys. They're really cute. And when I first, because I looked it up, I knew I'd published about these last year. Francis noticed that they look kind of feline, and they do look rather feline. It's cute little guys. But,
we've lost you, Jade.
Oh, you did?
Oh,
can you hear me now?
Yeah, I can hear you. Yeah,
Okay.
I mean, when I saw the picture of these wee foxes, I thought I want a herd of them.
Well, if you haven't seen the video, check. I actually haven't checked to make sure the video still works in the New York Times article that I posted, but there's a video in there, and they're so cute. I know. They'd make great little pets. I mean, if you know.
Well, I mean, if you're gonna do that, you want to get one of the Belyaev.
Well, yeah, but these are little guys. These are cute little guys. I don't literally mean they make great pets, because wild animals don't make great pets. It would be interesting to see if they did something with these little foxes like they did there in Russia, because they are awfully cute little things.
Yeah, the Belyaevs aren't wild though. That's the thing.
Well, I must admit, I'm looking forward to the next articles coming from this group.
Oh,
I really want to see the genetic analysis of these fox populations.
Well, they have this. They have the genetic. Some of the, at least here on this article that I posted.
Right now, where am I?
Okay,
that's the
Cell bottom
article.
Yeah, the Cell article.
I'll get it because I've got that many tabs open now. It's ridiculous.
I know. I'm having
getting lost.
Too. Oh, they even have a PowerPoint slide available. And they have, but this is open, so it's available. Let me see if I can post it. The outside chat. Yeah, I should have shut down a lot of my. There we go. I think you have to. Well, let me do it twice just in case. Okay, no, it's not working.
Right. Okay. Ooh.
Does that help?
It's interesting. Pulling up the first figure 1, and looking where Santa Catalina is clustering.
Mm-hmm.
Sort of.
There's even a, I think there's even a PowerPoint for this. There's PowerPoints for a lot of this. Damn it, system! I've got too much stuff open. Yeah.
Santa Catalina looks like it's out on a limb on its own.
Oh, it is. It is. I've been to Santa Catalina Island a few times.
Yeah.
It is
genetically.
Mm-hmm. Oh, yeah.
On that look as if they are out on a limb on their own.
Now, Santa Catalina is the only one of those islands that people visit on a regular basis, and people live there.
And that's the one where the tumors are found.
Right. It also has a herd of bison on it.
Why?
Bison.
Yeah, you didn't know that? Okay. Many years ago, I don't remember who it was, but at one time, some really rich person owned Santa Catalina Island. You'd have to look it up. I forget. And that person liked bison and decided to have some bison on the island. So they put some bison on the island. So Santa Catalina has a herd of bison on the island, and they've been there, I think, since the 1920s. I'm not absolutely sure. I have too many tabs open. Somebody else will have to look it up. And the bison have been there ever since.
Oh,
And just recently, someone was actually injured or killed by a bison, I forget, which is a very rare event because the bison are used to people. But whatever that person did, I don't know, people are stupid.
Here's an interesting little snippet from this. Each of these island populations represents a morphologically and genetically distinct subspecies, 4 of which, San Miguel, Santa Rosa, Santa Cruz, and Santa Catalina, have been listed as endangered under the U.S. Endangered Species Act after catastrophic declines due to predation by non-native golden eagles and introduced canine distemper virus. The individuals sequenced in this study were sampled in 1988, prior to the recent declines of these 4 populations. So they were already genetically distinct before they got hit by the canine distemper virus.
Yeah, this article, the Cell article, kind of strikes me as they need to get something out before they, their main interest is the cancer issue, right?
Right.
But in the meantime, they've got to keep publishing, and so if you're studying the genetics, you found a bunch of interesting things about the genetics. It's not the one you want yet.
I know.
But we'll get this article out.
Yeah.
But they were already, even before the population crash, they were already distinct, and they were showing monomorphism.
Yeah, that's
on a
interesting.
scale not seen previously in eukaryotes.
Let me correct something, by the way. I had that wrong. The bison were brought to the island in 1924 for the filming of a movie. The bison never made it into the movie, but because of cost overruns, they didn't take the bison back to the mainland. And currently they keep the population at about 150 animals, which is what it is now. That's really interesting. I thought, well, I do know at one time it was owned by a very wealthy person, and I frankly thought that person had brought the animals there, or at least a large portion of the island was. The other thing interesting about Santa Catalina Island is it used to harbor a species of dwarf mastodons.
Oh.
It's a formation of, if you know of island dwarfism. I don't know when the mastodons died off. Yeah.
Sorry, I'm just reading through this cell paper.
Oh, yeah.
And it's fascinating. Given that these were sampled in 1988, what year was the distemper started?
Like 91, I think.
So they were already, there was already genetic monomorphism before that.
Mm-hmm.
So imagine what the distemper did when it crashed the populations.
Yeah.
Oh, that is absolutely fascinating. So we do have genetic differences between these island populations of foxes. We already know there are those differences. Now we need, and that was before the distemper. Now we need to know what the genetic differences are now.
Oh, it did it again, damn it. It's not a mastodon, it's a pygmy mammoth. I always do that. I get the two of them mixed up. And at full height, they were about, like half the size of it. Actually, I'm not sure about that one. I'd have to look it up. But they weren't very big.
So, CRISPR, when do you think they will end up publishing their genetic studies on these?
I have absolutely no idea.
I know, because I don't know where they're at.
I mean, yeah, I have no connection to this
Yeah.
group, so I
No, but I
just
just wondered if
well, I mean, so this was published in late 2015.
Yeah.
So, I mean, this, it takes a couple years, to, I mean, depending on what you're actually wanting to do, what kind of confidence you're hoping to get to, everything else. So, I mean, and also, I mean, how many animals are you going to want to do the sequencing for? I mean, if you want to do the sequencing on let's say what, maybe five individuals per island should
Well,
get you a decent coverage, decent selection.
they took punch biopsies from a number of individuals from unaffected tissue, so they've already got the samples.
Yeah, I mean, but I mean, it's still $1,000 per genome. So
Oh, I know, but that is going to be fascinating when it comes out and we find out what is behind this difference, because it's not mites, because they all have mites. But anyway, I'm going to bring this to a close, peeps. So the answer to the question, what does the fox say, is keep an eye open for more papers coming out on the genetic diversity of these particular foxes in Santa Catalina in comparison to the other two islands that also have ear mites. And let's see what the differences are between these ones. So until then, I'm going to say bye-bye to everybody. And I'm going to ask everybody in here to say goodbye to the audience who has stuck with us for this length of time. Bye-bye, audience.
Bye, audience, that I was wrong. It's Santa Rosa Island, not Santa Catalina Island. It has the pygmy mammoths. Sorry about that. Bye.
Get your foxes neutered and spayed.
Yeah.
And on that note, I will just say, guys, let your balls swing low.